I'm really excited today to share some update progress that we are making at Sigilon. And so let's go. Let's start by really stating the problem. The amount of fear among patients with serious chronic disease is really profound. I know this as a physician that have experience over more than 25 years with lysosomal storage diseases, also with type 1 diabetes. Also, I know this as an executive working in some of the communities like rare blood disorders in my past 4 or 5 years. And as well, I know this as a patient myself, having type 1 diabetes for over 45 years. Tomorrow, we should be a source of hope. And I'm very glad that this whole conference over years are pursuing more and more innovative solutions to really help patients to transform their lives. So our technology is really to offer the possibility of a hope for all patients with serious chronic diseases. SLTX, which is Shielded Living Therapeutics, offer, I would say, an elegant way to harness the most powerful machine we have, the cell. Simply shielded cells from immune rejection and fibrosis, having steady and controllable protein secretion, not interacting with the host genome, having really straightforward manufacturing, redosable, retrievable, tunable, and durable over years. I will be telling you over the next few minutes everything about the product, how it works, what the levels we produce, and what the data we have. So let's go back a little bit. And as I said, the cells are very sophisticated, but the cells must work for us. And so since science 1980, or even earlier than that, most of the challenges on how to make cells working for us have been addressed. And I'm putting this series of publications, primarily by Bob Langer, which has been considered by, I believe, Harvard Business Review, Thomas Edison of medicine. He addressed many of these challenges in his publications, challenges like screening novel molecules to avoid fibrosis, showing the anatomic locations that would be preferable to have those cells being implanted, and many others. So this all and of course this work that he did together with Dan Anderson at MIT. So we leverage all of this information and create Sigilon. And we optimize some of this work, and we are here to deliver what we call implantable biological protein factors into the bodies. In this picture, you can see in non-human primates the spheres with allogeneic cells that have been implanted 4 months earlier. By the time of the termination, the cells were still viable. You clearly see that there's no signal of any fibrosis at all. And this was really one of the results that made us to move forward to deliver and to think about how can we do this into humans. So the Sigilon history, although short, is very, exciting. Again, all the work that was done in that MIT over more than a decade. And then last year we had a wonderful strategic collaboration with Eli Lilly. And we were very pleased that Lilly, among all other companies that they could have selected, they selected us to work and to find a functional cure for type 1 diabetes. And that is the goal of that partnership. We have grown the company to a point that we are now working with multiple programs. And we are target and on track to have 4 INDs in the next 2 years. And our lead program for hemophilia A is about to file IND by the end of the year, with first-in-human trials scheduled to go next year, first half of next year. We were also very pleased and happy that this program has received recently an orphan drug designation by the FDA. So let me introduce you the product platform, what we call the SLTX, Shielded Living Therapeutics, and each word has an importance there. So two components of the platform, the cell component, in which we have human cell lines that have been selected for safety, durability, and efficacy, and then we engineer those cells to produce the proteins that we desire in a steady state. We then encase those cells, and in each of the spheres, again, as you saw in the previous slide, the picture of the real spheres, they are 1.5 millimeters. In each sphere, you can put between 5,000 to 20,000 cells. So those cells were put in this breakthrough biocompatible matrix that not only shields cells from immune rejection and fibrosis, but also, and very important, maximize cell viability and durability. We then put these spheres with cells into the body through a very brief laparoscopic procedure. And of course, we are also testing other alternative sites for all the other programs that we want to achieve local therapeutic effects. But the intraperitoneal is the one that we have a wide distribution through the body. Those living factors or biologic protein factors produce the proteins that the patients are in need, and then I really consider indeed a functional cure. So this is a slide that we try to capture that Sigilon is really a modular platform. And since our founding, we rapidly optimize all of these components that you can see in the left of the slide with the characterization of the cell line, the optimization of the components of the sphere, and the standardization of the manufacturing process in order to how fast can we go to an IND. So the first program literally is taking us 2.5 years, and we believe that now that we are not only, we learn with this, but we are using like plug and play all of these approaches, we can be even faster, which is much faster than most of the other modalities, monoclonal, small molecule programs that you see from this concept to the IND. It's also very important that we have significant synergies across programs in manufacturing, in regulatory, and also in analytics. So that's the modularity is a very important component for Sigilon. Here, I am very proud with the team that we have assembled in Sigilon. The picture shows the executive team that really have an incredible, impressive track records of developing products and also creating value. And I'm very, really honored to be serving this board of directors with such prominent presence here that we have. In addition to that, I wanted to thank everyone in Cambridge, Massachusetts. We have now more than 90 people in our labs and office, and they are really working very hard, and they are extremely talented to really pioneer a new class of category of products to really help patients with their needs. So thank you guys over there. I know that you probably are seeing us through the webcast. So we have really greatly expanded our pipeline as our platform was maturing. And here you can see that the pipeline is really good on depth in terms of going really deep in many of the special therapeutic areas like rare blood disorders, lysosomal storage disorders, as also as a breadth in terms of going to diverse therapeutic areas that you can see with other chronic disorders and metabolic diseases as well. Today, we'll be focusing on the ones that I have the blue line. I hope that in the next meeting, we'll be reporting the many progress that we are making on the diabetes type 1 program that we have with Lilly. But today we'll be concentrating those 5 other points. Let's start with hemophilia A. And here is a market that I'm so happy to be working again in hemophilia A after many years working there, learned a lot. Particularly, I understand the challenge of the current treatments, and I think that the flat dosing, and you see this in the left graph, where we demonstrate a steady state for the plasma human factor VIII levels. This will have profound impact on the clinical outcome. In the graph on the right is just to exemplify that if you take US and EU4, UK, France, Germany, and Italy, we can have more than 20,000 patients eligible for our platform. The regulatory path has been really clear. We have really very good meetings with all agencies in UK, in Germany, and the US. We have received, as I said, orphan drug designation, and our enabling studies are ongoing, and we are targeting first in humans next year. Very quickly, we have very, I would say, enthusiastic and a lot of good feedback that we had from hematologists as also from the patient communities in hemophilia. We present many of the impressive results we had in the past 12 months, and in the next 12 months we'll be presenting even more. Why we share with gene therapy for hemophilia A the same concept of a really steady PK levels of a factor, and this is a very important element to get positive outcome. We have some differences, and I want to highlight just a few. One, eligibility will not have any impact of antibody to a virus. Also, in terms of the range of the factor rate will be much tighter, as well that we are not going to modify any patient cells, and we can be redosed multiple times, and even retrieval is possible. So the clinical trials, I don't need to spend time just to say that this is exactly copy and paste any of the gene therapy trials. We don't want to invent where we don't need to invent. So clear endpoints, clear definitions for the trial. We said about hemophilia A, another area that we are working is on not only hemophilia B, but also I wanted to show one picture here on the factor VII deficiency. And this is a very interesting picture that, different from the current product in the market, Novo7, our product is not an activate product, so the activation will happen in the body by and when it's needed. So it really would work very well in the prophylaxis, and there is no other gene therapy or any other program in pipeline in any other company for these patients that are really suffering a lot. And very importantly, some of these patients may cost more than a couple of millions of dollars per year due to the very short half that they have in terms of Novo7. So just shifting gears, and I see the clock going very fast, on the lysosomal storage disorders, we have a very exciting results on both Fabry disease and MPS. In Fabry, as you know, we have a deficiency on the enzyme GLA that accumulate substrate levels, GB3, in an elevated way. And here you can see that after 2 weeks of being implanted cells into knockout mice, we can clearly see that there is a reduction of the substrate very remarkably. And I was very happy that we saw this reduction not only in plasma, which is kind of easy, but also in other tissue levels, which is not so simple to happen. So this proves that our technology could really have a big impact on patients with this condition. Very quickly on MPS 1, we have exactly the same results where we produce cells producing human enzyme iduronidase. And in the upper set of the small graphs, you can see that even lowest dose of spheres containing cells can produce very dramatic effect that is comparable to the ones that we have in the lower set of graphs where we show lauronidase, which is aldurazyme. And again, we are very similar to clinical doses of aldurazyme, which is very exciting, and we are moving those 2 programs into IND very soon. So I don't want to and probably will go another 30 seconds, but we have also tested some local compartments of the body, and in here we have implanted spheres producing anti-VEGF antibody. And as you can see in this model of rabbits, that we have induced the breakout in the leakage in the vitreous with the VEGF, and when you apply the spheres secreting anti-VEGF, in this case we are literally secreting Lucentis, you can see a reverse of the problems that we had. Very exciting, and we are going to have and to show more data in the second. So finally, we are very really putting a priority to have a process that is cost-effective and is scalable, and we believe that we are in a good way to really have our cost of goods go into the order of monoclonal antibodies, which will be a very different thing. So Sigilon is really going to pioneer a new class of drugs, dramatically changing patients' lives with serious chronic disease. I thank you all for your attention. Thanks.